Hi friends, this is Miss Medico channel, and today we are going to discuss bacterial sex genetics. We had already discussed a transformation, transduction. Now we will discuss about the conjugation. This is actually a part of bacterial sex genetics. It's actually the bacterial sex at its best, and that is sexual union occurs at its best by this process that we call conjugation. In conjugation, what happens is that the uh, DNA is transferred from one bacterial cell to other bacterial cell directly. So the DNA is transferred from one bacterial cell to other bacterial cell directly uh, by cell to cell contact. In conjugation, there occurs cell to cell contact. From one bacterial cell, DNA is transferred to other bacterial cell through the cell to cell contact, and this results in an extremely efficient transfer or exchange of genetic information from one bacterial cell to another bacterial cell. And this exchange can occur between unrelated bacteria also, not among species, but it can also occur between unrelated bacteria. And some major mechanism for the transfer of antibiotic resistance. So in conjugation, what happens? DNA is transferred from one bacterial cell to another bacterial cell through cell to cell contact, and it's an efficient mechanism by which we can transfer DNA from one bacterial cell to another cell. For conjugation, what happens? For conjugation to occur, one bacteria must have this one bacteria must have a this is the bacterial chromosome. One bacteria must have this structure that we call self transmissible plasmid. So for the conjugation process to occur, plasmid, one bacteria must possess this circular DNA, this double stranded circular DNA that is extra. For conjugation, one bacteria must possess this self transmissible plasmid, also called F plasmid. F for fertility, because it helps the bacteria. F for fertility, not other word. So for the conjugation to occur, one bacteria must possess a self-transmissible plasmid, that is also called F plasmid. F is for fertility. What is that plasmid? Plasmid is circular. Plasmid is nothing but it's a circular double stranded DNA molecule that lie outside the bacterial chromosome. And this plasmid carries many genes, including those for drug resistance. And F plasmid encodes the enzymes and proteins necessary to carry out the process of conjugation. Bacteria that possesses this plasmid are called F positive bacteria, means plasmid positive, are donor bacteria. And bacteria that does not possess this plasmid, they are called F negative bacteria. also called recipient bacteria. So we have two types of bacteria, two kinds of bacteria between which conjugation can occur. We have F positive, that means those bacteria which possesses this self-transmissible plasmid, and those bacteria which receives this uh, self-transmissible plasmid. So we have two types of bacteria between which this conjugation can occur. This what happens? The self-transmissible plasmid has a gene that encodes and the proteins and enzymes and form a sex pens. It encodes the proteins जिसके पास सेल्फ ट्रांसमिसेबल प्लाज्मिड होगा, वो उसके पास एंजाइम्स होते हैं, जीन्स होते हैं. दे encode the enzymes and proteins and form this structure that we call sex pens or piles. This is the main structure by which conjugation can occur. The self-transmissible plasmid, what it has genes that encode enzymes and proteins and that form this structure that we call sex piles. This long protein protrudes from the cell surface of donor bacteria and binds to, I will show you here, this protrudes to the cell surface of bacteria that has got the plasmid. This sex pilus protrudes and gets attached to the cell surface of other bacteria that does not have that plasmid. And actually this sex pilus forms a bridge between these two between the donor bacteria and recipient bacteria, and that bridge is called a conjugal bridge, between which transfer of this plasmid takes place. So conjugal bridge is formed. What happens? A nuclease enzyme is there, breaks off one strand of a plasmid, means it gets breaked. I'll show you here what happens. Sorry, this is like this. So nuclease enzyme is there that breaks the one strand of the circular DNA, and this circular DNA gets transferred through this conjugal bridge to recipient bacteria. As one strand is passed, other strand, as shown by the dotted line, is formed by other nucleotides over there. तो यहाँ पे भी डबल स्ट्रैंड बनती है न्यूक्लियोटाइड से जो वहाँ पे होते हैं. इस तरह से प्लाज्मिड बनता है. सो as one DNA is passed through the conjugal bridge to the other bacteria, new nucleotides are paired with this already strand. So at the time of sexual union, conjugal bridge break होता है. उसके बाद conjugal bridge breaks and both bacteria will now possess, conjugal bridge breaks, both bacteria possess now plasmid at the end of the sexual union. So this happens in conjugation. It's an extremely efficient process of genetic transfer from one bacterial cell to another bacterial cell. I hope you have understood the video. If you have any query, comment in the comment section. Thank you.